Now let's look at the eukaryotic cells. Eukaryotic cells are present in many domains, and I will write down the names of all these domains that we will talk about. They are present in mainly protists. Protists are your amoeba, paramecium, euglena. The domain fungi, which could be yeast, or it could be the mushrooms, plants, and animals. So the kind of organisms that we are more familiar with are all eukaryotic organisms. Eukaryotes are different from prokaryotes because firstly they have a nucleus, and they also have many other true organelles. True organelles are those that have membrane around it. So when under a microscope, when a true organelle is seen, usually different regions can be identified. Think of the prokaryote as one big studio apartment that can function with all its components, and the eukaryote is more organization. As the cells picked more functions to do, the cells started to differentiate regions within the enclosed environment, and what started was compartmentalization, where certain structures which are called as organelles arose, and they're very specialized to perform specific functions. Eukaryotes, like prokaryotes, they also have a plasma membrane, and we will see that what are the other structures that are present within the plasma membrane. But keep in mind, compartmentalization is one of the key factors for the eukaryotes to be more complex. Compartmentalization is also helping to increase efficiency of many reactions. For example, a mitochondria would be one organelle that is used for the powerhouse of the cell. A chloroplast would be another organelle that is used to manufacture nutrients, and that is usually in plant cells only. Other organelles that we will see later on, for example, nucleus, the administration and the control of the entire cell is controlled by the nucleus. The concept is that the prokaryotes were the ancestors, and there's a hypothesis, which is the endosymbiotic hypothesis, that is shown in this diagram over here. The original prokaryotic cell had a little bit of its own DNA. Remember, we said earlier that it was like an extra chromosomal DNA was in the nucleoid region. And for example, this is an example where we can see that the cell has engulfed an organelle, and in this particular example, mitochondria and chloroplast are shown over here. What the cell, as you can see over here, is reaching out and invaginating its plasma membrane to absorb this or to ingest this bacterium inside it. And when they ingested the bacterium, it became a part of its own structures that are present within the cell. And this is endosymbiosis, which means it benefits both parties. The animal cell benefited because the mitochondria provided its its energy, or the powerhouse. The mitochondria, which was a primitive bacteria, was only focused in creating energy and did not have to worry about getting the nutrients. So over invagination of the plasma membrane have created these structures and the endomembrane system, which helps explain that how mitochondria and chloroplasts were once upon a time independent prokaryote, and they took up living inside a eukaryotic cell. So what organelles are present in eukaryotes? You can see the list is very long. Nucleus is a region which is shown in the center over here. The most visible region within a cell is the nucleus. Now for this class only, we are showing you an image of an animal cell over here. This is not a plant cell. This is only an image of an animal cell. The plant cell, you can look in the book. It's a slightly different, and I'll show you the regions where they are different. But 156 is mainly focused on animal cell and its function, so I'm focusing on the animal cell only. Mitochondria is another organelle that is present over here. These are all mitochondria. Looks like a kidney bean. The endomembrane system is a very extensive system, and it's a system that starts from the nucleus and keeps on invaginating and all over, and we'll talk about this system in another slide. Lysosomes and peroxisomes and vesicles, one S on a vesicle, vesicles and vacuoles, these are all structures and sacs that are present within the cytoplasm, and some of them are for recycling and very specialized functions, and again we're going to do that later on. Now the only one that is not mentioned over here is the chloroplast in this picture, because they're only present in plant cells. Besides these organelles, there are other structures found in the eukaryotes. Now we call them structures because they do not have a complete membrane-bound definition. Those are ribosomes, cytoskeleton, centriole, cilia, and flagella. And in the next coming slides, we will look at each one of these. It's very important that you can identify this diagram. If I give you this diagram in a test, I expect you to be able to recognize each region and know what it is. I'm not giving you the names over here right now, but as we study it, I think it will be more easier to understand. Now, however, over here I want to give you two terms. One is the term called as cytoplasm, and the other term is cytosol. Cytosol is more like the liquid, the water, the liquid. So if you think of the room that you're living in, the floor would be the cytosol. Cytoplasm is a term that is used in which we say all these organelles are suspended in the cytosol. So let's go on with the first organelle. Let's look at it in more depth, and this is the nucleus, the most conspicuous one when you look under the light microscope. Generally, we show that a cell has one nucleus, but it's not uncommon to find more than one nucleus in a cell. The nucleus contains chromatin. Chromatin is the thin elongated strands of DNA, and it's present in the fluid of the nucleus, which is called as the nucleoplasm. So this would be the nucleoplasm, which is the fluid of the nucleus, and within the nucleoplasm all these fine I don't know if you can visualize see it, these dark lines that are shown over here are the chromatin. Chromatin is thin, and it is composed of DNA strands. Now, only during cell division, when the cell is ready has replicated and is ready to divide, the chromatin starts condensing, and when they start condensing, they form more rod-like appearances, and these rod-like appearances are the chromosomes. So chromosomes are always going to be thick, and chromosomes are associated with dividing cells. So when the cell is dividing, you will find chromosomes. Now in a cell that is existing all the time, you will always find chromatin. It does not. So chromatin and chromosomes are two vocabulary words, and you should know the difference between the two. The nucleoplasm is the semi-fluid or the matrix that is present in here. All of this is enclosed by a nuclear envelope. The nuclear envelope pretty much holds this the content of the nucleus, and also separates it from the cytoplasm, which is the region that is over here. That's the outside region. Within the nucleus, this should not be the word nucleotide. It should be nucleolus. So make sure you fix this over here. Nucleolus. Nucleolus is a dark region, which is a region where this region over here is the nucleolus. Nucleolus is a region which manufactures or synthesizes ribosomes. And we'll talk again about ribosomes. I mentioned earlier about the ribosomes being able to manufacture proteins, but ribosomes are present in the cytoplasm. But however, they are synthesized in the nucleolus. On this nuclear envelopes, you see these little openings, and these openings are the nuclear pores. The nuclear pores are pretty much openings that will help the DNA, which will pass its message, or it will help the communication from the nucleoplasm to the cytoplasm. And we'll get into more depth as we go on in this class. Okay, the